Hi everyone. In the immunology subject, we are going to discuss about a topic called types of immunoglobulins. Yes, we have discussed what is an immunoglobulin, what is the structure of an immunoglobulin and the functions of the immunoglobulin in different parts. Now, in this part, we are going to discuss about types of immunoglobulins. First of all, what is a immunoglobulin? How do we define it? So, just recollect it. So, immunoglobulins are immunological active proteins or active serum proteins, which are also called as antibodies. Yes, which are also called as antibodies. So, this is what we have learned in the structure of antibody or structure of immunoglobulin in another part. In this part, these different types of immunoglobulins, we are going to discuss each one, its biological properties and structure in detail. So let's begin. So the plasma proteins are majorly classified into two types. One is going to be called as albumins and second one is going to be called as globulins. And these globulins are again classified into three types of globulins called as alpha globulins, next beta globulins, gamma globulins. And these are the gamma globulins from which we are going to get our immunoglobulins. So our antibodies or immunoglobulins are gamma globulins. Okay, and depending upon the heavy chain of antibody or immunoglobulin, the immunoglobulins are going to be classified or differentiated into five types. So what are those five types? IgA, IgM, IgA, IgD and IgE. So again, these are going to have the subtypes also that we will discuss in detail now. The Tislayers in 1937 separated the plasma proteins into albumins, globulins, what I told you, all beta and gamma. So albumin, what is an albumin? So here, this is what? It is a protein present in the highest concentration which contribute to the colloidal osmotic pressure of a plasma and acts as a non-specific transport proteins. Those are albumins. Then what are globulins? These include specific transport proteins, clotting factors, and then complement system and inactive precursors of certain hormones like angiotestinogen, like that. Okay, and in these gamma globulins acting as a circulating antibodies, that is especially coming to the gamma globulins. So these are going to act as circulating antibodies which are very important in our specific immune response, which is already known as immunoglobulins. And these immunoglobulins, as I said, depending upon the amino sequence of heavy chain, they are being classified into five types. IgA means which type of heavy chain is there? It's gamma. IgM means the heavy chain is mu type. In the IgA, the heavy chain is alpha type. In the IgD, the heavy chain is delta type. And in the IgE, the heavy chain is epsilon type. And each immunoglobulin class differ in its general properties, its half-life time, its distribution in the body, and interaction with other components of the host defense system. So that's the reason why we are going to read about all these things individually now. So let's begin with the first one, IgG. So here is the immunoglobulin G or IgG, and the structure of human IgG is going to possess four polypeptide chains. That how we have read about the structure, in the same manner you are going to have it. So if you look at this one, then you will get, this is the structure of an IgG, and this is going to be a relatively more stable in its nature or in its structure. And how many polypeptide chains are there? There are four polypeptide chains. Okay, and these are all going to be linked with one another by disulfide bonds. Okay, and the heavy chains are going to be interlinked with again disulfide bonds. And one more thing that you have to remember here is the light chains are going to be either kappa type or lambda type. And how many variable regions and how many constant regions are there? There are one variable region in all the 4 polypeptide chains. And coming to the light chain, it is having one variable region and then one constant region. And heavy chain, one variable region and 3 constant regions. How many are there? 3 constant regions are there. Okay. And then coming to its what we call it as terminals. One is going to be of N terminal, another one is going to be the carboxylic terminal. Okay, and this is the most predominant class of immunoglobulin, that is IgG is the most predominant class of all immunoglobulins, and it is going to be of approximately 70% of the total immunoglobulin in human serum. Okay, and normal serum concentration of IgG is about 8 to 10 milligrams per ml, and it is the greatest amount in internal body fluids and provided particularly during the secondary immune response. Where? In the secondary immune response. And the molecular weight of IgG is 1,50,000, and its sedimentation coefficient is 7s, and it has a half-life period of 2 to 5 days. So this is what you have to remember. Its half-life period is only 2 to 5 days. And there are 4 antigenically distinct subclasses of IgG, namely IgG1, IgG2, G3 and G4. Okay, so these are the subclasses of IgG. Under the electron microscope, IgG looked like a Y-shaped one. So that's what we have seen there. The carbohydrate content of IgG is about only 3%. As it is a protein, it also contains a little amount of carbohydrate, and that is only 3%. Okay, so this is all about the IgG that you are supposed to write. The structure of human IgG is resembling the Y shape, and it is relatively stable for polypeptide chains. The light chains are made up of kappa or lambda type, and 2 heavy chains are made up of gamma type. So here you have to write the heavy chains are gamma type, and one variable region and one constant region in the light chain, and in the heavy chain one variable region and 3, how many are there? 3 constant regions are there. Okay, that is CH1, CH2 and CH3, and this is the predominant class of all other types of immunoglobulins, and they are about 70% of the total immunoglobulin, and they are going to be of about 8 to 10 mg per ml of the serum. And if we are going to take its molecular weight is 1,50,000, and sedimentation coefficient is 7s, and its half-life is only 2 to 5 days. And we are having the 4 types of the subclasses of IgG, that is G1, G2, G3 and G4. And then coming to the biological properties of IgG, which is very important. So IgG is the only immunoglobulin that crosses the human placenta and thus offer a passive protection to the newborn baby for about 6 to 9 months. So remember here, IgG, G indicates the gamma heavy chain. In the same manner, gynec also, you can remember the gynec also starts with the G. So that's how which immunoglobulin is going to pass from the mother to the fetus through the placenta means, don't get confused, it's a gynec related. So IgG, it's a trick, just you can remember it. And now this IgG activates the classical complement pathway during the antigen antibody reaction. That is second one. And the third one, this IgG is going to neutralize the toxins and viruses that are present in our body. And even that, it will also bind to bacteria. Okay, and IgG opsonizes, that is opsonization is a mechanism that is going to occur. What is that? IgG opsonizes them, thereby enhancing their phagocytosis and elimination, and this process is known as opsonization. Okay, opsonization. Now this is all about the IgG immunoglobulin. So it is the most predominant immunoglobulin. And then moving to the second type of immunoglobulin, that is IgA. That means here the heavy chain is alpha type, and it is found only to a small extent in the serum, but predominantly seen in the external serum mucus secretions of respiratory, gastrointestinal, and urinogenital tracts. Even IgG is the second most abundant among all the immunoglobulins. That is, it is about 10 to 15% of the total percentage, whereas IgG is 70%, and here it is 10 to 15% next to IgG. And there are two subclasses of IgA that exist, namely as IgA1 and IgA2. The IgA1 has both interchain and intrachain disulfide bonds, whereas Ig2 is completely devoid of it into chain disulfide bonding between the heavy chain and the light chain, and the two chains are held together by non-covalent linkages. So these all, so we had two types of IgA, that is serum IgA, and then second one is the secretory. So by looking at the figures, you can have like have an idea of it. Okay. So coming to the serum IgA, so this is a secretory IgA. Okay, serum IgA is a monomer having a structure very similar to our IgA. How you are going to have your IgG in the same manner, IgA is also going to have, and it has 4 polypeptide chains, 2 light chains, 2 heavy chains. Okay, and the light chains may be of kappa type or lambda type. So here you can see this is second type of the thing. So only one will be there in the serum IgA. The alpha chain of IgA differ from the gamma chain of IgG. So that is a heavy chain is going to differ. There it is in the IgG, it is gamma type. IgG, it is gamma type, and here it is IgA, which is alpha type. And its molecular weight, that is IgA molecular weight, is going to be of 1,60,000, whereas that is 1,50,000. And the sedimentation coefficient is both, that is 7s. And here IgA half-life period is 6 to 8 days, whereas our IgG is only about how many days? 2 to 5 days. And here it is 6 to 8 days. And serum IgA has more amount of IgA than the IgA2. Okay, so we are going to find the more serum IgA in the body. Then coming to the secretory IgA, the second type. Secretory IgA is nearly an equal mixture of both IgA1 and IgA2. So whereas in the serum IgA we had only IgA1 is more, but in the secretory IgA we are going to have both IgA1 and IgA2 50-50%. Okay, and human secretory IgA is much larger molecule than serum one. It is having a big structure because it is going to be of like this. That's why it is big in nature, and its molecular weight, as it is mixing of two things, it is of nearly 5 lakhs. And it occurs as a dimer. So you can see here it is a dimer, two monomers linked together by a secretory component with the J chain. So that's why this is going to be called as secretory IgA antibody. And both the secretory piece, so here you can see the J chain and as well as the secretory piece, are going to be the proteins. Okay, and the secretory piece is synthesized by the local epithelial cells. So the epithelial cells are going to release some sort of secretory component that is going to attach with this dimer, and this IgA dimer is going to get this stabilization and the proteolysis nature, and it is bound by disulfide bonds and non-covalent bonds to the Fc region of the IgA dimer. And the secretory piece has a single polypeptide chain of a molecular weight nearly 70,000. That means only this secretory component is going to have how much molecular weight? 70,000. And the J chain, the link that is going to be present in between these two, J chain, is also a polypeptide chain of molecular weight 15,000. So that means here, if you are going to clearly observe, this is a secretory component. Yes, shaped one is a secretory component, and here you can see the J chain. And this J chain is also a polypeptide chain with of a molecular weight 15,000, and the secretory component is going to be of about 70,000 molecular weight. So this is about the secretory IgA, which is a dimer containing the IgA1 type and IgA2 type in equal proportions. Okay, then we are going to move to the biological properties of this IgA together. So IgA is termed as a mucosal paint or antiseptic paint of the mucous membrane. That means these IgA antibodies are going to be painted on our mucosal membranes, and that's how we are being protected. And it is believed to inhibit the adherence of the microbes. So it is going to be the first line of defense mechanism. Okay, and even to our respiratory, gastrointestinal tract, urinary genital tracts, thereby preventing them from colonizing and invading the different organs of these tracts. And even IgA does not fix complement, but it is found to activate the alternate complement pathway. So we are having the two types of complement pathways, which we'll discuss in another part, complement system. So we had two types of complement system. One is the classical and alternate pathway. So here this IgA is involved in activating the alternative complement pathway, and that's how they are going to neutralize the local toxins, promote phagocytosis, and activate bacteriolytic activity. But this occurs only in the presence of an enzyme called as lysozyme. And IgG is also found to produce immunity against some tapeworms also, that is protozoa elements, okay, parasites. And even this IgA is present in the colostrum, protects the baby from intestinal pathogens. As we classified the serum and secretory IgA, so obviously this is going to be present in the secretions of mother milk. So this IgA is going to protect that enters into the baby through the breastfeeding, reduces the gastrointestinal infections in those babies or infants by reducing the entry of pathogens of the environment, which otherwise occurs of bottle feeding is as follows. So that's the difference between the bottle feeding baby and the breastfeeding baby, is going to be the difference of having these IgA immunoglobulins. So that's how the doctors, everyone prescribed to have a breastfeeding is because of having these IgA immunoglobulins, which are giving the protection to the baby against the pathogens. Okay, so next moving to the third type of the immunoglobulin is Igm. That means the heavy chain of this immunoglobulin is mu type, which is a very important type. And here IgM is the largest of the immunoglobulin. So if the question is which is the largest immunoglobulin, so blindly you can say that it is IgM. It is often referred to as a macroglobulin. So if you see the structure, so this is the IgM structure. See how many are there? 5 are there. That's why this is the largest immunoglobulin, and it is going to have a molecular weight of about 9,50,000, whereas our IgA is going to be of approximately 1 lakh only. But here we are going to have a 9,50,000. So, and there are polymers of how many immunoglobulins? 5 immunoglobulins, and each with the 4 polypeptide chains. And these are all going to be connected with a single J chain in the how we have seen that in the IgA. In the same manner here also you are having a J chain. Okay, and all the Fc portions are connected with one another and stabilizes the group. Now the serum level of IgM is very low, that is about only 5 to 2 mg per ml. And this low serum level is due to its short life period. That is, it's having only 5 days and a low synthetic rate of 5 to mg per kg body weight per day. Okay, so that's the reason why they are going to be of very limited in the blood serum, means they are having a very less lifespan, and the carbohydrate content is more. Obviously each one is having a 3%. If altogether means we may have the above 5 only, no, 5 to 8, it's of for example each is going to be 2, then 5 to the 10 will be there, so 10 to 12% of carbohydrate content is there. Then what about the sedimentation coefficient? For IgG and IgA, the sediment coefficient is 7s, but here it will be a little bit more because it's a pentamer. So obviously it is 19s. The sedimentation coefficient is 19s. Okay, and there is no subclasses for IgM. So for IgG we have seen the Ig1, Ig2, IgT, Ig4. In the same manner we had gone through the second type IgA, we had IgA1 and IgA2. But for this IgM we don't have any subclasses. And this IgM is the earliest immunoglobulin to be synthesized by the fetus. Okay, it is not going to be transferred from the mother. The fetus itself is going to be synthesizing the first immunoglobulin, that is IgM, by starting about the 5th month of the fetal life. Phylogenetically, IgM is the most primitive immunoglobulin present in all vertebrates from Lampreys onwards. A monomeric form of IgM is found on the B lymphocytes. Yes, B lymphocytes. Then coming to the biological properties of IgM, this is the first antibody to appear in the primary immune response. This is very important. So which immunoglobulin is going to be appear in the primary immune response means obviously IgM. Okay, and it does not persist for long because of its short life that I told you, less than 5 days. So therefore it is going to be used as an indicator of recent infection. So if your blood serum is going to test and found the IgM in the blood serum, that indicates you had a recent infection. Then the maternal IgM does not cross the placenta, but the human fetus can synthesize IgM. If its B cells are antigenically stimulated, as in congenital infections. Okay, most natural antibodies, such as the natural classical groups of ABO blood groups, is having this IgM. Then coming to the next, by having the multiple antigenic binding sites, because it's a pentamer, isn't it? So obviously it is going to have, each one is going to have 1, 2. That's all. How many? It's a multivalent. So 10 are there. So 5 to 10 is going to be the antigen binding site, and IgM has high functional affinity for having the multivalent antigens. But its multivalency, it can also inhibit the mobility of the pathogen and enhance their phagocytosis. So that is more arms, more hands are there, more you can work. In the same manner, the antibodies, the 5 immunoglobulins are put together as a pentamer. So obviously it is having a capacity of holding the multivalent antigens also, and that's how it enhances the phagocytosis by macrophages. So it is also involved in such type of opsonization, then complement fixation, then agglutination, and even in the cytolysis. So as I told you, because of its macromolecular size, it is primarily localized in blood and thus protects against the blood serum infection. So mainly involved in the blood serum infections, whereas the IgA is going to be the mucosal paint of our mucous membranes and acting as a first line of defense. And then moving to the next type of the immunoglobulin, that is IgD, whose heavy chain is going to be of delta type. And here the two heavy chains are of delta type and the two light chains are going to be either kappa type or lambda type. And it is slightly larger than the IgG, having a molecular weight of 1,80,000. Whereas IgG is 1,50,000 and here it is 1,80,000. The larger size of the IgD may be associated with the extended region of the hinge region. What is the hinge region? The central flexible part of the immunoglobulin is called as hinge region. So here you can see it. Where is it? So at last. So here it is. IgD. This hinge region is going to be of more in size. So obviously that the reason why it is having a little bit high molecular weight when compared to the IgG. IgD is limited in the blood serum. The average serum level is only of about 0.03 mg per ml. So this is why it is very limited in concentration. Okay, and then the average sedimentation coefficient is 7s. So as it is have a 4 polypeptide chain, all the things, it is 7s only. And IgD is rich in carbohydrate. That means it have more quantity of carbohydrate. and the rate of synthesis is 0.4 mg per kg body weight, and its half-life is only 2 to 3 days. It is also having very less span of life. And there are two subclasses of IgD, that is IgD1 and IgD2, and it is formed associated with the surface of B lymphocytes, that is antibodies or B cells. Then moving to the biological properties of IgD, IgD has not been shown to have any antibody activity, and it does not mediate any effector functions attributed to immunoglobulins. The only one main function is it is found to be associated with the B lymphocytes along with the IgM is found to act as an antigen receptor. That's the only function that we can see with this IgG. Okay. Then moving to the last type of the immunoglobulin, that is IgE. That means here the heavy chain is going to be of epsilon type, and IgE also possess 4 polypeptide chains, 2 light chains and 2 heavy chains, and the 2 heavy chains are epsilon type and the 2 light chains are either going to be the kappa type or lambda type. And this is the only immunoglobulin which does not have a hinge region. This is very important. This is not going to have the hinge region, and its molecular weight is about 1,90,000. And it is a heat labile immunoglobulin. Labile means it is a very heat sensitive, whereas other are going to be the heat stable immunoglobulins. What are the other? IgG, IgA, IgM and IgD. And these are going to be present in very minute traces, that means 0.0004 mg per ml. Suppose if this ratio is very high, its concentration is high in the blood, then that condition is called as eosinophilia condition. Okay, and high levels of AGE is going to be encountered mainly in the intestinal helminthic infection. That means the persons who are having this some sort of a cold or intestinal helminthic infections, then we can have the more number of IgE antibodies in the blood circulation, and their half-life period is about 2 to 3 days, and they are going to synthetic rate of about 2 to 3 micrograms per kg body weight per day. And here also the carbohydrate content is more, that is about 12%. Altogether the IgM is going to of about only 10 to 12, though it is a pentamer, but here IgEC the singly it is of 12%, and its sedimentation coefficient is 8s, whereas IgG and IgA is 7s, but here it is HS, and IgD also 7s only, whereas your IgM is 19s, sedimentation coefficient is 19s. So it has a special attraction towards the mast cells and basophils which bear a receptor for this IgE. We have discussed in the cells of immune system about this one. And IgE is usually known as skin sensitizing antibody, and originally it was named as a reagin, okay, which is involved in causing the allergic reactions. And coming to the biological properties, obviously we have very few. High level of IgE in children with shows that they are going to be with the manifest infection with protozoans, okay, that is worm infestation, seem to have a protective role. Then IgE antigen interaction on mast cells result in degranulation of our mast cells with the release of some sort of chemical substances like vasoactive amines or histamines, okay, which cause the allergic reactions, and IgE mediates the reagenic hypersensitivity, that is type 1, which is also called as anaphylaxis. So the types of hypersensitivity will also be discussed in another part that you can go through it. Now let's have a quick overview of all the 5 types of immunoglobulins that we have discussed, and their physiochemical properties and the biological properties. So let's have a quick review of all these things. So here are the 5 types, and here are the physiochemical properties. So sedimentation coefficient, as I told you, all are having IgG is 7s, IgA is 7s of first type, but it is another security is a dimer where it may be of 15s, and IgM is 19s, IgD is 7s, and IgE is 8s. Then the molecular weight, as we discussed, is 1,50,000, and here it is 1,62, even the 5 lakhs in the dimer case, and then IgM is 9,50,000, and IgD is 1,80,000, and IgE is 1,90,000. The number of basic 4 polypeptide units, here it will be 1, here it will be 1 again, but here as a dimer it may have 2, and IgM is a pentamer, so obviously 5 are there, and IgD is 1, IgE is also 1. Then coming to the light chains, all are having kappa plus lambda type, but heavy chains, IgG contains gamma, IgA is alpha, IgM is mu, IgD is delta, IgE is epsilon. And then coming to the property of valency for antigen binding, here the valency will be 2 because it's a monomer, and here as a monomer it is having 2, but as a dimer it is having 4. And then as a pentamer, the 5 are there, so 5 to 10 valency of antigen binding site, then again here 2 and here 2. And the serum concentration is going to be range 8 to 16 of IgG, then IgA is a 6 to 4.2, and IgM is 5.2, and this is going to be of about, that is 5 to 2, then it is of 0.03, and IgE is even traces amount, it will be there 0.0004. okay, and half-life periods are going to be like this: 25, 6 to 8 days, 5 days, 2 to 3 days, and 2 to 3 days. Then coming to the carbohydrate content, we know that it is 3%, then it's 10%, it is 10 to 12, even these two are 12%. And then coming to the biological activity, antibody activity will be positive except in the IgD. Whereas in the cross placenta, that means the which immunoglobulin is going to cross the placenta means only one globulin, that is IgG, gynec. Okay. Then reagent activity, that is hypersensitivity activity, will be seen obviously in the IgA, which is also called as reagin. And then complement fixation can be seen in two types of immunoglobulins. One is going to be of IgG, another one is IgM. Then which immunoglobulin fixes the macrophages? Obviously IgG. Then which immunoglobulin fixes the mass and the basophil cells means? Obviously it is IgE. So these all about the what we call it as immunoglobulins comparison with of biological properties and immunological properties. So these are all the different types of immunoglobulins, their biological properties. So this we end our types of immunoglobulins in detail. Okay, thank you.